All right, I hit the record button, but nothing's happening. Okay. Well, it says it's recording, but I don't see any red light.
It's recording on my screen.
It looks like it here. There's a blinking red light.
Okay, that's good. It's probably somewhere. Okay. All right, let me get rid of all this. Give everyone a couple minutes. So we went from 22 participants to like 5. But it's okay.
I know that great, but they let me know.
No, I get it. I mean, it's aggravating. For me, I can't even imagine what you guys are going through. So thank you. I really can't even imagine what you guys are going through.
And it's too bad because it seems really interesting. I would like to be able to do the stuff in the lab to see what things are actually doing, the growing and stuff. I can't quite picture it all the time.
No, that's the really fun part. It really is going from lecture right to lab and then doing stuff. And the labs really only take an hour and a half. They're fun, and you review everything from lecture. So like I said, I apologize to you guys. I feel bad. I'm really trying to do the best I can for you guys. I don't know if other teachers are doing Zoom meetings or whatever, so I really am trying. So bear with me.
I liked your the YouTube things you did for like using the little discs, for the zone of inhibition and those sorts of things. Those are good.
Yeah, I wish you guys could do your own antibiotics.
I mean,
a lot more fun. But anyway, I mean, you're probably never going to do it in the field. It's just kind of cool to do it. You get a better understanding. All right, let me clear everything out and we'll get started. Did I leave off with like Neisseria? Let me
pull out my notes.
Yeah.
It's supposed to be admit all. I don't understand. More.
And then the clinical diagnostics, the gram stain, CSF.
For meningitis. Meningitidis. Yeah, well, you're going to see they're going to be in the macrophages. Let me just, this is supposed to automatically admit everybody. So I'm afraid to allow, switch to unmute. Disable the waiver. All right, so I literally have a Zoom account now. I bought it, so this should go smoothly. All right. All right, so we are recording?
Yes.
Rumor has it. Okay, all right, so is this gram positive or gram negative, Neisseria?
Positive.
Wrong.
I thought it was negative.
Ew.
It is negative.
Damn it.
Okay, so Neisseria gonorrhea is a leading cause of gonorrhea and meningitidis. Okay, went over this. All right, so just kind of remember it has fimbriae. All right, so gonorrhea, once it gets into your mucous membranes, it's going to attach. All right, and the other key thing with this is it cleaves immunoglobulin A, which is in all of your mucous membrane. So this is, once it gets in the mucous membrane of wherever, it couldn't be in the eye, it can be, and not to be crass, but you can get gonorrhea in your throat. All right, you can get it anywhere there's a mucous membrane. We don't have to get into the details. All right, but it cleaves the secretory IgA and the mucous membrane. So we have no way of tagging this. So it's a virulence factor, makes it more virulent as a way of evading the immune system. I think I mentioned it's strictly a human infection, so this you really can't get from a fomite. And I said, jokingly a little bit, you can get it from sharing toys, not to be crass, but you can get it from sharing latex toys, right? Infectious doses 100 to 1,000. So, kind of remember you have to have 100 or more of these to even make it virulent enough to get into your membrane and be able to cause an infection. And once again, these microbes, just because they are in your body, if they don't find conditions good, they're not gonna populate. They're not gonna colonize, right? Right. And I think we talked about this with males. They're 10% symptomatic. In females, are 50%, 40 to 60% symptomatic. So a lot of these people don't even know they have it. And I think the other day I talked about people having this, not knowing they're carriers, and then once again, it gets cleared up, and then people get, we call them frequent flyers in the healthcare system. They just go out and reinfect themselves. Right? Okay. I think I talked about this. So it's going to cause all kinds of fibrotic scarring and inflammation. It can lead to inflammation of the vagina, ureters, the Salpinges, and it causes a lot of ectopic pregnancies, right, which are extremely painful, and they sometimes can be fatal. Right? Okay. Right. And then gonorrhea in the newborns, as it's passing through the birth canal, they can get this. And then prophylactically, I think they pretty much still use silver nitrate, right? Because remember, the conjunctiva is a mucous membrane too. So, this is where we ended, correct? Right around here, maybe? Okay, let me just make sure.
Yeah, I have that written down, this information.
Okay, good, all right. So they're going to be inside the neutrophils. The neutrophils are going to go in, and if they can get them on their own, because they're not super tagged with the immunoglobulin, they're going to engulf them, all right? And remember, these are double D in shape, right? They're going to be gram-negative. Right. So I'll just go over meningitis again, and I think I mentioned this is very, very severe, very, very fast-going, and this is in the cerebral spinal fluid. So it has to find it somewhere from the nasal passage and get into the blood, or somehow it gets into the blood-brain barrier. All right. And do you guys know what phagocytic cells you have in the blood-brain barrier? Like what defense mechanism do we have if something's in the blood-brain barrier?
Is that like the glial cells?
Yeah, actually the glial cells, the astrocytes, the glial cells. All right, very good, excellent. All right, we have those. They're really supportive cells, right? But this also has a capsule, so even if they capture it, they're gonna have a problem getting through it. It has those fimbriae again. Once again, it has a protease, and this has an endotoxin. You guys remember anything about endotoxins with gram-negative bacteria?
It has that LPS.
LPS.
Yep.
These are endo, and these are gram-negative. So these get released upon death, that LPS, lipopolysaccharide. And like I said before, it causes major inflammation and intravascular coagulation, fever. It can be pretty brutal. So what are the signs and symptoms when they give you a case study on something? What are going to be some signs or symptoms, or how are they going to phrase meningitis to you? Robert, what do you think?
Rob, excuse me.
Sorry, I didn't mean to put you on the spot there.
No, that's all right. I ate these salty chips and they were bad. It was real bad. Definitely fever.
Fever, yep.
Nausea, vomiting, maybe.
Okay.
Horrific headache.
Yeah, horrific headache. It's going to be sudden onset, horrific headache. And remember, there's a lot of inflammation going on in your cerebral spinal fluid. And I think I mentioned, if I didn't, I'm mentioning again, your brain is not pain sensitive. So it's going to start swelling that dura mater, that pia mater. It's going to cause all kinds of swelling and stretching that capsule. It's extremely painful. So the patient's going to be grabbing their neck. It's got nuchal rigidity, like the back of the neck. And they're generally going to be in flexed posture, Koenig sign. They're going to be flexed posture, knees up. And if you try to straighten them out, it's going to exacerbate the pain. They're going to scream. Right? So they're going to be curled up. They're going to be in flexion. They don't want any tension on that nervous system, on the central nervous system. And so they'll phrase something like that on your board exams or whatever. There's always usually a case on that. All right, so there's going to be sudden onset. And this is something serious. So if somebody is diagnosed with it, your children or someone in college, this is usually something with close proximity because you're sharing saliva or something. It's in the mucous membrane. So if it's in a dormitory or whatever, they're going to give everyone prophylactic antibiotics just in case.
Isn't there a vaccine for it?
Yes, there is. Okay. So yeah, so usually human reservoir nasal pharynx, higher institutional setting. So, it could be prison, group home, dormitory, wherever, where you're going to be in constant contact with people in close proximity. Right? Just looking over that. All right. Oh yeah, we went over this. So there's a picture of it. All right.
Oh.
All right. So what do you think that is?
Necrosis.
It is, but what caused it?
The red blood cells being laced.
Yeah, intravascular coagulation. All right, so everything clotted in the blood in the capillary bed, so we can't even feed this tissue. So it just starts dying. It's hypoxic. It can't get nutrients. You can't run the ATP pumps. All right, so you have hydropic change. All right, so the cells can't. This is probably beyond the scope of the course, but I'm going to mention it. I won't test you on it, just so you know. You don't have any ATP. These cells can't produce energy. They can't run the pumps. So sodium keeps going into the cell. They can't pump it back out. Water follows that, right? Water always follows sodium. These cells explode. They die, causing necrosis and gangrene. Well, gangrene is if Clostridium perfringens is in the area. It's going to start eating on that dead tissue. We'll go over that towards the end here. All right. So we're going to gram stain the cerebrospinal fluid. You're going to do a lumbar puncture below the spinal cord. All right. So some books will say L3, L4, L2, somewhere lower. All right. And it's going to be in the blood. Nasal pharyngeal sample. They're going to swab that. Culture for differentiation. All right. And then really we're going to do rapid tests. We're going to do an immunoglobulin test immediately. They'll know what's going on. All right. Treatment, like I said, I'm not going to ask you guys any specific antimicrobials. I don't think that's something you'll learn in the field. I don't know if it's really part of micro. You'll probably have to take, you guys have to take pharmacology?
No.
I
No?
think we do. I think we do for nursing.
Oh,
Yeah,
do it?
They're going to ask.
Oh, yeah, we do.
Yeah.
Yeah.
I'm working with, I'm in the conversation with the school so that I can offer it so you guys don't have to go to KU or whatever. I'm working on that.
Mhm.
All right, and yes, there is a vaccine, Robert, contains specific purified capsule antigens. They're going to actually take part of the capsule, those antigens, those HLA markers, and they're going to take part of that, attenuate it, inject it into the patient, and we're hoping their dendritic cells will present that to the immune system like we talked about in Unit 3. So I'm just going to go over some of these real quick. They're not going to be that clinically relevant in this area. Von Hamela, right, found in the nasal pharynx. Right. Usually somebody, it has to be opportunistic. They have to have cancer, diabetes, or they have to have a diminished immune system before this is going to really take effect. I'm not going to test you guys on it, but you've heard it. Ciniobacter can cause infections. They almost look like mold or fungus. You might see that if you guys are going to be working at the VA or possibly at ECMC. You might see that. I'm not sure. Right. So, okay, that should be the end of 19, correct? I think I'm asking you.
Correct.
No, sorry. Duh. All right, hold on. Let me just switch over.
I try to follow along with the PowerPoints that you put up. So
I know. Well, yeah, but do you get anything to play on your computer?
no.
Really?
I did yesterday. I was studying and I was able to get some of them. Yeah.
And the PowerPoint, if you click on,
Yeah.
they're right.
Yeah.
Okay, good.
Mhm.
I thought maybe.
No, mine. I mean, it could be. I do my work. I use it at work, so
Mm.
maybe the work computer, but it's
Yeah,
usually pretty good.
They might have a whole bunch of malware, like at ECC.
Yeah, I would.
Well, now we used to have 7 layers of security. Now we have 8, apparently.
Well, if somebody didn't get through the first 7.
Oh my God, I can't even download anything with with a 7-digit number. Like they're like, oh, it's a screen number. I can't do it. All right. So let me get over this. Like I said, we're in really good shape. I think we have technically 6 more classes and maybe 1 more unit. So I'll do these. Maybe we can do like an hour and a half or whatever, because I know 2 hours is pretty strenuous for you guys. So maybe I'll cut these back and get them recorded and up, and then I'll give you guys like a week or 10 days to get everything done. Their course technically ends August 12th, but I'll leave everything up. So if you guys run into an issue or time management problems, they'll all be up. I have your lab midterm done. I'm going to post that. I'll give you guys about 2 hours to do that. It's all going to be on just clinically relevant stuff that we would have learned in lab, nothing we haven't covered in class. And I have your next test. Did it post for you guys? Exam 2?
Yeah, I'm pretty sure it posted.
Okay, so it's
I
only
haven't tried it yet, but
I think it's only on like 9, 11, and 12. It's in there. So I didn't do a lot. So that should be easier. And then the other tests should be shorter, more compacted, so they won't be nearly as bad. All right. So let's get over the rest of this. So these are also going to be gram-negative bacilli. So these are going to have that LPS. Remember, they've got that outer membrane, that polysaccharide, peptidoglycan layer, and then they have the other inner membrane. So these are generally, in most cases, a little bit harder to kill, because we have to get the antimicrobial through that outer membrane, and we know membranes decide what comes in and out of a cell, whether it's a human or a bacteria. Then it has to get through the the other layer, and then it has to go through the initial membrane around the bacterium itself. Okay, let me get some. All right, does anyone need to get into the participant 6? Do I have to admit anybody? Okay, hold on, somebody's waiting. Abby is, sorry guys. My bad. So these are aerobic gram-negative non-enteric bacilli. So what does that mean, non-enteric?
Does that mean it's not coated with
hmm.
fimbriae? I don't know.
Nope. Okay, so anything that's enteric, whenever you see that, it means it's usually in your digestive tract. All right? So these are generally non-enteric.
Oh, so like if you have an aspirin that's enteric coated, is to prevent it from digesting too fast, like in your digestive system?
Well, yeah, enteric coated. Yeah, we want, well, yeah, so let's say I have an antimicrobial and it's designed for enteric. So we want that to be able to go through your stomach, which is high acidity. We want it to be able to break down in the duodenum or the small intestine, where the actual infection is. Perfect. Or they have like enteric coated aspirin back in the day. Yeah, so we want that to get through the digestive system, because there's only 2 things that get digested in your stomach. Do you guys know what those are? One of them hopefully you're old enough to know about, which is alcohol. The other one is aspirin. So if we have it enteric coated, we want to bypass it. We want to make sure it gets through your stomach and then it gets into your intestines. All right. So whenever you see enteric, they're telling you it should be part of your microbiota, or it would be found in your digestive tract. So if you were in lab, we'd be doing a whole bunch of tests on these, and these are the tests here, more or less. We'd be doing all these biological tests in tubes, which is a little bit passé or old school. They don't really do that anymore, but it's something that what's going on? That they still they're still on board, so we still do it. All right. So these are a large, diverse group of non-spore forming bacteria. So if they're non-spore forming, they're probably generally easier to kill. All right. All right. So what would be spore forming in your digestive tract that we're going to be talking about coming up?
C. diff.
C. diff, Clostridium difficile. So Clostridium, all the Clostridiums are going to be spore forming. right? And that's why Clostridium difficile is so difficult, because we're killing out all their competition. So now all we've left is spore formers, and we know we can't kill those, right? So they're going to just vegetate and just reproduce over and over again. That's why we have such a problem with C. diff right now. So they have wide ranges of habitats, large intestines. So right there, enteric means it's going to be in your digestive tract. Zoonotic, hope you guys know what that is. All right, so that is generally something that is in a mammal or a reptile or a bird or an amphibian that generally wouldn't be in our intestines. All right, so we've all heard of salmonella. It's on eggs and it's on reptiles and everything. That usually isn't a problem for chickens or other zoonotic animals. It's a problem for the humans, right? These could be in your respiratory tract. They're in the soil, obviously, from feces or excrement, and they're in the water. So when they close beaches or whatever, we know if there's, if they close Woodlawn Beach, we know the coliform. Right? One would be the enteric bacteria. And if the enteric bacteria is elevated, we automatically know there's going to be E. coli, because that's one of the major components of feces. Most are not medically important. Some are true pathogens. So when they say true pathogen, that means we could be totally healthy adults, and these would cause disease in most healthy adults, a true pathogen. Right? And then some are opportunistic. So remember, opportunistic means they usually wouldn't be a problem, but you've got something going on, the rest of your microbiota is wiped out, or you're immune compromised, or your immune system's fighting something else. So now they take the opportunity to infect you. Normally, your immune system would get rid of them. Right? All have an outer membrane of lipopolysaccharide of the cell wall. This is an endotoxin. Remember, this is released when they die. This is that LPS, lipopolysaccharide. Okay. Constitute large group of human bacterial pathogens due in part to lipid A and the bacterial cell wall. All right, so I'll be going over this a few times, and there will be questions on your exam. All right, so I'll give you basic questions, and I'm not doing this to be mean. I just want you guys to get prepped for your boards. Like, I want you all to do well in micro, but my job here, in my opinion, is to get you ready to take a board exam that really counts. All right. So here's the thing. Any gram negative, it has that lipid A or LPS, lipopolysaccharide, triggers fever, like Robert said before, vasodilation. That's going to cause your blood vessels to dilate, inflammation, shock, and disseminated endovascular coagulation. All right. So I'm going to ask you guys a question again. What component of inflammation is going to cause shock? Anybody remember?
Wouldn't that be a blood pressure drop?
Right. Why? Why is that?
Because there's not enough blood pumping through the heart from the, I don't know, I'm done.
No, you're on the right track. Remember, inflammation, vasodilation. We want the capillaries are going to dilate. They're going to, capillaries are only one cell thick. They're going to dilate. They're going to spread apart. All that fluid's going to leave the capillary bed, go into that interstitial fluid that's bathing all of your cells, which is the inflammation. So that's what happens. We have such a huge vasodilation and inflammatory process. That fluid literally leaves your capillary beds, and now you have no blood volume to pump. All right, your heart's pumping. So if you have no blood volume, if you have very little volume, your heart's going to pump very rapidly to get whatever you have to feed the tissue. I can't think of the word right now. But yes, we wanted to perfuse the tissue. God, I'm getting old. All right. Most gram-negative bacteria that breach the skin and mucous membranes grow at 37 degrees. So remember, this has to breach, cut, has to get in your mucous membrane somehow. Grows at 37 degrees Celsius, which is your internal body temperature, and can invade the immune system and cause disease in humans. And some of these we're going to see, literally, I said, they're going to want to trick your macrophages and monocytes and some of your neutrophils to literally engulf them. they want that to happen because they know their immune system can't really touch them very easily. And now they can travel anywhere they want and get off the Uber whenever they want to. All right, so I put this in here. So lipid A, that endotoxin, LPS, all right, it's going to just cause all these things. So just kind of remember, it's going to trigger fever. It's going to damage your blood vessel walls, right? Right, disseminated vascular coagulation. This is where the blood clots in your capillary beds. So remember, if it clots in your capillary beds, everything distal past that can't get blood supply. Right? And your blood supply is how all of your cells get all of their nutrition. Right? There's no FedEx or Amazon Prime to get things to your cells. It has to go through your cardiovascular system. So if we're talking about diabetics, when they destroy their capillary bed, that's why their toes turn green. They have all that neuropathy because the capillary beds are destroyed in the distal extremities. The nerves die and this tissue starts dying. Causes major inflammation, and this is what causes the shock. Plasma loss and reduction of blood pressure causes that shock. Okay, so aerobic, gram-negative, non-enteric bacteria. So this is stuff that shouldn't be in your digestive tract. I'm not going to say that some of us don't have Pseudomonas in our digestive tract because it's everywhere. Okay, do you guys remember me saying anything about Pseudomonas before?
I thought it smelled like grapes. Is that what you said? That's about all I remember.
See, I can't even smell the petri dishes in front of you, and you guys. One girl said, "Are you bold?" I go, "What are you talking about?" She goes, "She's bold. You smelling that?" I'm like, "It smells like grapes. It's delicious." Don't lick the petri dishes. But yes, Pseudomonas it usually is green or blue, and sometimes it grows. Like if you go into the bathroom, you see green stuff on the fixtures. Okay, that's generally not going to be mold. That's usually Pseudomonas. Oh my God, it's telling me to upgrade. Can you guys see that?
Oh yeah, it says remaining
literally
meeting time,
opened
9
an account
minutes.
Tuesday night. Son of a. Okay, I'm gonna take a deep breath and deal with this. I don't have to restart, guys. All right, so anyway, Pseudomonas doesn't need carbohydrates to live. It will grab any carbon source at all. Anything that has carbon in it, it can live off of it. And Burgdorferi is going to cause some opportunistic infections. All right, we're going to see that. Brucella and Francilla are zoonotic pathogens, so those come from other animals. Bordetella pertussis. You guys ever heard of that vaccination?
Mm-hmm.
All right.
Yep.
That can be deadly. All right. And usually it comes from, a parent or a grandparent carrying it in their throat. For us, it's not a problem, but infants and small children are a problem. Legionnaire, we've heard of Legionnaire disease, mainly a human pathogen, but that really has to live near amoebas. So it's got to be around water. So when you guys go to Wegmans or Whole Foods or Trader Joe's or whatever, and they're misting all those vegetables, just saying. Okay, so Pseudomonas is aerobic bacilli, right? Ubiquitous in soil, so it's everywhere. It is decaying organic matter, so it is a bioremediator. We want this. It starts breaking down things in the soil, organic matter, dead, whatever, and likes moist environments. Problematic in hospitals. This loves wound victims and burn victims. And it's opportunistic pathogen. So it's generally, if your immune system is relatively good and you're relatively healthy, isn't that much of a problem. The problem right now is Pseudomonas is getting immune to a lot of our antimicrobials, so it's becoming a major problem. So small gram-negative rods with a single polar flagellum. All right, so it has a flagellum. What can we deduce about this right now?
It can move.
It can move, all right? So if it's in a wound or whatever, or it's on a stitch or it's on a pick line or something, it is mobile, right? It's free living, primarily in the soil, seawater. It doesn't like seawater as much because of the salt concentration. Freshwater also colonize plants and animals. Important decomposer and bioremediator. So we want this. So if we have an oil spill or some kind of, we want this in our landfills and stuff. We want it to start breaking things down. with contaminants in home and clinical settings. So, like I said, I wish you guys were in lab. I'd have you go home and swab your sponges, your can opener, you know, around your faucets or whatever, and then we could streak it out and we could actually see it growing. We'd streak plate it, grab that colony, and then grow it, and it would smell like grapes. You'd say, oh my God, I just had Pseudomonas growing on my sponges. It was growing, you know, wherever, on my bottom of my purse or wherever. It's all over. Use aerobic respiration to do not ferment carbohydrates. So remember this, it can't digest carbohydrates. All right, so it's going to take any carbon source, anything that has carbon in it. It'll use it for food. It's pretty amazing. Produce oxidase and catalase. So what do we know about that? So if it can produce oxidase and catalase, it can deal with oxygen. All right, so the ones that can't produce this, we can hit it with oxygen, it'll kill it. Most produce water-soluble pigments. That's why things turn green or they have blue. Some Pseudomonas is red, but they really make pretty colors, honestly. All right, so I'm getting nervous. I'm going to take a break here and pause this. I'm going to restart this. I don't know why I upgraded. I swear to God, I did it Tuesday. And they told me I was good to go. All right, so I'm going to stop the meeting and then restart it. Everyone okay?
Okay.
All right, sorry guys. I am trying.
Fine.
All
Okay.
right. All right, let me stop video and stop sharing.